Hello everyone, and welcome to today's webinar, Learning from Hospital-Acquired Infections, Understanding Current Reprocessing Regulations, Product Validation, and Clinical Human Factors, brought to you by Nelson Laboratories in partnership with QMed. I'm Lynette Fan, Client Services and Delivery Specialist at UBM Americas, and I'll be your moderator today. We have just a few announcements before we begin. This webinar is designed to be interactive. Each widget within the dock at the bottom of the console will open in a different window for you to view additional resources, speaker bios, and an overview of the webinar. There are several social media widgets for you to like, share, and talk about the content with your peers and communities. At the end of the program, we will ask you to complete a short survey. Please take a moment to fill this out. Your feedback will provide us with valuable information on the subject covered in this webinar and how we can improve future events. You can also launch the survey at any time by clicking on the survey button at the bottom of the console. The slides will advance automatically throughout the event. You may also download a copy of the slides by clicking on the PDF labeled Download Slides, located in the Additional Resources widget. If you are experiencing any technical problems, please visit our webinar help guide by clicking on the Help widget. Now, onto the presentation discussing today's topic will be Emily Mitzel, Consulting Manager at Nelson Laboratories. Emily, I now hand it off to you.
Thank you, Lynette, and thank you for joining us today. First, we will be talking about some news articles that have just recently come into the news of when it goes wrong in healthcare settings. We will then get into written justifications for tests, sample numbers, and acceptance criteria. Next, we will be discussing validations of cleaning, disinfection, and sterilization of reusable medical devices. And then we will touch on human factors associated with this type of testing and the reprocessing of medical devices. A USA Today news article from a few weeks ago stated that more than 8 healthcare facilities have had outbreaks of hospital-acquired infections. This article specifically blames automatic endoscope reprocessors, stating that reprocessing instructions were followed. One thing to make sure is that the automatic endoscope reprocessor must be validated for each scope type that is reprocessed. In this CNN news article, 7 hospital patients at Ronald Reagan UCLA Medical Center were infected with carbapenem-resistant Enterobacteriaceae, otherwise known as CRE, from October to January. 2 of those patients have now died. The device that caused the HAAMI did not have specific FDA clearance. The company made modifications to the device to make it safer, but did not revalidate. According to the British government, drug-resistant bacteria could kill 10 million people a year and cost up to $100 trillion by 2050 if their rampant global spread is not halted. It is also noted that in Europe and the United States alone, around 50,000 people currently die each year from infections caused by bacteria such as E. coli. Even the president has gotten involved. According to the CDC, 2 million illnesses and 23,000 deaths from antibiotic-resistant bacteria each year in the United States. The president is implementing a 5-year national action plan at a cost of $1.2 billion. New rapid tests to detect emerging resistant bacteria will be developed and researched for new antibiotics and vaccines. The most recent and important guidance document came out from the FDA a few weeks ago. The draft of this document came out in May 2011, and it has been a very important document for medical device manufacturers and healthcare facilities. Major differences from this document versus the draft document are specified in Appendix E, which includes devices for which a 510K should contain data to validate reprocessing instructions. It also requires 2 quantitative markers for cleaning validations and specific definitions of controls that are utilized in the cleaning validation. So a hospital-acquired infection, or more broadly phrased, a healthcare-associated infection, is an infection whose development is favored by a hospital environment. This infection is acquired by a patient during a hospital visit that was not in the patient prior to the time of admission. 7% of the people in developed and 10% of the people in developing countries will acquire at least one healthcare-acquired infection, and hundreds of millions of people will be affected by healthcare-associated infections worldwide. And this leads to a significant mortality and financial losses for healthcare systems. There are many meetings and standards that have been come about in the past few years to combat healthcare-acquired infections. In 2011, the AAMI and the FDA put together a medical device reprocessing summit. Also in that year, the FDA guidance document on reprocessing came out, like I mentioned earlier. There are also two other very important documents that came out that year, and that is the AAMI TIR 30 and AAMI TIR 12. In 2013, AAMI and FDA held a roundtable discussion, which I facilitated, our main topic of that was how clean is clean and how to make sure that we are performing the testing correctly. Just last fall, two other documents came out, the AAMI TIR 34, which is the water for reprocessing of medical devices, and TIR 55, which includes human factors engineering for reprocessing medical devices. Also, just last week, ST91 was finally submitted, and that is a document for flexible and semi-rigid endoscope reprocessing in healthcare facilities. So this is specific for reprocessing scopes in healthcare facilities, which we all know is a very timely standard at this point. The healthcare facility has also been combating healthcare-acquired infections, and these specific instances include special drying cabinets for clean scopes, ethylene oxide sterilization equipment for scope sterilization. Just a few weeks ago, the CDC issued a surveillance protocol, which I'll be talking about more later in this presentation. They've also been putting together proper training and credentialing, which is very important to make sure that their people are trained properly to reprocess medical devices. It is also important to follow the standardized instructions for reprocessing and having standardized inventory to ensure that the reprocessing instructions are appropriate and easy to follow. The reprocessing study timeline starts with a very in-depth written justification for the contamination, cleaning, test methods, and acceptance criteria of the validations that need to be performed. Next, the cleaning, disinfection, and sterilization validations are then performed, and then a summary report is written with all of the results and the acceptance criteria showing that the acceptance criteria was met. We find that the majority of questions asked by FDA is not due to incorrect testing, but incomplete justification. Cleaning depends on 4 criteria. They include device design, device compatibility, instructions for use, and human factors. One very important standard is ISO 17664. It states a validated method of cleaning shall be specified and include a description of accessories required, the identification of all water qualities that are utilized for each step of the process, the amounts of chemical residuals that are tested after the cleaning process is performed, and also information regarding the temperature, concentration of solutions, and exposure times, and all of the techniques included that are utilized in the cleaning procedure that includes rinsing. One of the first things to consider is the device condition when performing the cleaning and sterilization validations. As we probably know, a new shiny device will be able to be cleaned and sterilized easier than a device at the end of its use life. FDA has required simulated use cycling, and the number of cycles and the simulated use cycling depends on the type of device. So we want to get the device into more of a use state prior to performing the validations. Another couple of very important aspects of the cleaning validation are clinically relevant test soil and worst-case testing. Those are a couple of buzzwords that FDA are very, very focused on to make sure that the testing is performed correctly. I presented this information to the FDA in 2011 and have never been questioned on our test soil since. On the left shows all the ingredients that may be utilized in test soils, and the right column shows why the ingredients are clinically relevant. So we take these types of ingredients and put together a clinically relevant soil that is exact for the simulated use of the device that will test the exact surgical procedure that is utilized for the device. FDA now recommends the specific test soils we utilize for validations for other companies that they have. Probably the most important part of cleaning validation are the contamination steps. In the contamination, we want to ensure that we are simulating the surgical procedure exactly. So we want to ensure that we are immersing the device, handling the device with bloody gloves like the physician would do. We are performing actuation of the device as it would be performed during the surgical procedure. Then we need to ensure that the device is held in the test soil for the amount of time that the surgery would take place. The final part of this is to ensure that the device is dry, the soil of the device is dried onto the device, as we need to simulate the time that it would take for the device to move from the OR suite to the central processing area to be cleaned. Here are some examples of how the contamination takes place. We can either fill the fluid pathway with a blood soil, spray the soil on, handle the device with bloody gloves, and we can also soak the device, or a combination of these depending on how the device is utilized. Here is an example of what we do not want to see in the clinical setting. As we know, some surgical centers sometimes get backed up with many trays, and there are some surgeries now requiring more than 15 trays, and there sometimes may be longer wait times for these to be reprocessed. The central processing area can get very overwhelmed with the number of devices they need to reprocess, and so we need to ensure that the cleaning instructions for these devices are easy to follow and maintain throughout the area. After the cleaning procedure takes place, visual inspection is necessary. A few years ago, the only visual inspection that was specified was performing visual inspection with a naked eye under normal lighting conditions. We have now found with issues of arthroscopic cannulas from 2009 that we need to ensure that we are looking at the inside of medical devices to ensure that they are clean also. This can be conducted by a boroscope, and we also utilize magnification to look at different types of parts of devices that cannot be seen very well with the naked eye, including screws and other types of areas that are hard to reach. Here's a picture of a human tissue from an arthroscopic cannula that is seen by a boroscope. So you can see that there's human tissue that's retained in that cannula. After visual inspection takes place, the device is then extracted and tested for specific residuals. The types of residuals depend on the surgical procedure itself and also where the device will be, where the device will be sold. The U.S. FDA requires two quantitative methods of testing, and those are the most popular methods of testing right now are protein and hemoglobin testing. In Europe, specifically for the CE mark, Geobacillus stereothermophis spores are specified as necessary to test for medical devices. If the devices are going to be sold or utilized in Germany, and specifically the Bavarian government, Enterococcus faecium needs to be performed as well, the bioload reduction. Other tests that may be necessary include cytotoxicity to ensure that there are no detergent residuals left on the devices, as well as endotoxin testing if the device comes into contact with cerebral spinal fluid or brain tissue. Here are some examples of acceptance criteria for the specific residuals that may be tested. Most of these come from TIR 30 and are specific from a study from an endoscope. These are the numbers that have been accepted by the FDA for quite a while now, and if a medical device manufacturer wants to specify different acceptance criteria, they can. It's just that they need to perform testing and justification that would be appropriate to change these numbers. Next, we'll be discussing disinfection instructions and validations. Again, ISO 17664 is an important standard regarding disinfection validations, and a validated method of disinfection shall be specified. Items that need to be included are all the accessories required, the contact time, temperature, and concentration of the disinfectant, the specific water quality that is required for specifically the final rinse, and that should be more of a critical water supply. We also need to determine if there are any chemical residues left on the devices after the rinsing is performed. The disinfection acceptance criteria is based on Spalding classification. Critical devices must go through a high-level disinfection, and the acceptance criteria is a 6-log reduction of a mycobacterium species. For semi-critical devices, those devices will go through an intermediate level disinfection, and the acceptance criteria of this disinfection includes a 3-log reduction of a mycobacterium species and a 6-log reduction separately of each of 4 vegetative organisms. And these organisms include Escherichia coli, Klebsiella pneumoniae, Staphylococcus aureus, and Pseudomonas aeruginosa. Non-critical devices need to go through a low-level disinfection, and the acceptance criteria is also 6-log reductions separately of the 4 vegetative organisms that I just mentioned. We are seeing a move from the FDA that these non-critical devices also include any type of non-patient contacting devices that are located in the OR suite. So we need to ensure that any type of instrument and equipment that is located in the OR suite can be properly cleaned and disinfected to ensure that we are not performing any cross-contamination between patients and physicians. Sterilization is important to ensure that terminal sterilization of the device is necessary. The most important methods of sterilization in the healthcare facility are steam, ethylene oxide, and vaporized hydrogen peroxide. VHP sterilization has become even more popular as it is easier to use than ethylene oxide sterilization. Again, the standard ISO 17664 has a lot of information regarding what is needed for the validation of the sterilization cycle in the healthcare facility. Information needed are all the accessories required, and these include the BIs, biological indicators, the chemical indicators, the packaging, etc. We need to note the concentration of the sterilant, as well as the humidity, temperature, pressure, and exposure time. And we also need to note any post-sterilization techniques that need to be performed, specifically as an aeration step for the ethylene oxide sterilization. To perform a sterilization validation, we first perform a sterility assurance level validation with half-cycle parameters. Next is the dry time validation with full-cycle parameters, and then temperature profiling of the entire case to ensure that every part of the case is getting up to the correct exposure time temperature. The overkill method is utilized for the sterility assurance level validation since the bioburden of the device will change based on different patient use and also the environment of each healthcare facility. Due to this, we are testing for the half-cycle time to kill a sterility assurance level of 10 to the minus 6. In this testing, biological indicators are utilized that have a known population and resistance. Biological indicators are placed in the most difficult to sterilize areas. These include lumens, mated surfaces, and cannulas. In a tray that is utilized for, say, a hip replacement surgery, we may have up to 20 to 25 different sutures of different types of biological indicators in spots that are most difficult to sterilize to ensure that everything in the whole entire case is sterilized appropriately. FDA also requires 510K numbers to be utilized for sterilizers and all accessories, including the biological indicators, chemical indicators, and packaging, and that standardized sterilizer parameters are utilized, and no more extended cycles are performed. The parameters for these can be found in AAMI ST79, TIR 12, and the new FDA guidance document. Here is an example of what those standard cycles look like. In the U.S., the most commonly used cycle is 132 degrees Celsius, and the sterilization exposure time is a 4-minute exposure time. The most common parameters in the EU are 135 degrees Celsius with a 3-minute exposure time. Many of our clients need to validate both of these parameters as they market to both of these areas. We have found that a lot of trays will pass through a combo sterilization cycle of these two parameters, in which we use 132 degrees Celsius and a 1.5-minute half cycle time. Therefore, our clients can test both of those cycles in one sterilization cycle, thus decreasing their time of testing and their expense. Another item that I want to touch on is end-of-life testing. We need to know when a device is unacceptable for use. FDA asks many manufacturers for this information. This is mainly performed by a certain number of steps, which depends on the device itself. So this may include simulating the surgical procedure, performing manual, automated cleaning, disinfection steps, and sterilization steps. The number of cycles depends on the device itself and how long it'll be before the device goes into the end of its use life. As it is very difficult for a healthcare facility to specify how many times a device has been utilized and reprocessed, the device manufacturer normally writes down a lot of the information how to determine when the device is at its end of use life. Human factors considerations plays a main role in reusable device testing, as the sterile processing personnel are the ones that perform this testing, or this cleaning, disinfection, and sterilization. And it is very important for them to perform it correctly to ensure that the healthcare-acquired infections are at the very minimum amounts. There are many standards and guidance that are focused on human factors, and I have added them into this slide. There's many AAMI documents, as well as FDA guidance, CDC guidance, and also ISO guidance. Just a few weeks ago, the CDC came out with a surveillance protocol for surveillance of duodenoscope products. These include that a surveillance needs to be performed every 4 weeks to annually, or by a certain number of procedures per year or per month. What this includes is that a scope will be utilized on the patient, it will then be reprocessed, and then will be cultured to ensure that the reprocessing instructions have been followed and are appropriate. This scope will be then taken out of use as the testing is performed, and that scope will be tested for high and low concern organisms. This is important to ensure that healthcare facilities are reprocessing duodenoscopes properly. There is a summit at the FDA just next week that will talk about scope reprocessing, and will talk about the surveillance process of duodenoscopes as well. We have been performing these validations, or these scope surveillance studies for years for different manufacturers and clinical facilities. Today I will end this talk with discussing FDA's 6 criteria to follow that are very important for medical device manufacturers and healthcare facilities to ensure that the proper reprocessing instructions are followed, and that we are limiting and decreasing healthcare-acquired infections. In these 6 criteria, the labeling of the devices should reflect the intended use of the device. Reprocessing instructions for reusable devices should advise users to thoroughly clean the device. They also should indicate the appropriate microbiocidal process. They should include technically feasible devices and accessories that are legally marketed. All of the instructions for reprocessing should be comprehensive and also should be understandable. There's a lot going on with reprocessing medical devices right now, and a lot of reference standards that are associated with this type of testing. And it's very important to read through them all and understand how the testing should occur and how the healthcare facility should reprocess devices. Our Nelson Labs website also has a lot of references and specific information regarding cleaning validations of reusable medical devices, disinfection validations, sterilization validations, and device life cycle testing. Thank you for your time today, and now we will turn the time back to you, Lynette.
Thank you, Emily, for the valuable information on learning from hospital-acquired infections, understanding current reprocessing regulations, product validation, and clinical human factors. For upcoming webinars and for the archive of this webinar, please visit qmed.com. This webinar is copyright 2015 by QMed. The presentation materials are owned by or copyrighted by Nelson Laboratories. The individual speaker is solely responsible for their content and opinions. On behalf of QMed and our sponsor, Nelson Laboratories, thank you for attending and have a great rest of your day.